Hello everyone, I am Yasi Ayub and we will try to understand together the Coombs test. Coombs test or antiglobulin test is an immunologic test which is used to screen or to prevent many immunologic diseases mediated by antibodies. By realizing the Coombs test, we try to know if there is or not in a selected serum specific antibodies that can recognize specific antigens. These antigens are on the surface of red blood cells, the binding of antibodies on antigens, and then the fixation of antibodies by anti-antibodies will make red blood cells agglutinate, creating a visible clump that will prove the presence of these antibodies. The Coombs test was first described in 1945 by Robin Coombs, a British immunologist that studied veterinary medicine, became a professor and researcher at the Department of Pathology of University of Cambridge, and then founded the British Society for Immunology. To make the Coombs test possible, we are going to add Coombs reagent to a fluid that must contain red blood cells with antigens on their surface and specific antibodies of that antigens. The Coombs reagent is a fluid that contains anti-immunoglobulins. It means that it contains antibodies that can recognize and fix human antibodies. But how can we make it? In reality, we simply take human blood, then we extract its serum. This serum contains different human proteins. We take from it the human antibodies and then we inject them to an animal. The immune system of this animal will consider human antibodies as foreign antigens and will develop a humoral immune response, which will allow synthesis and secretion of anti-immunoglobulins by plasmocytes. So the Coombs reagent is no more than antibodies that can recognize and bind to our human antibodies. In practice, we have two Coombs tests, the direct and the indirect Coombs test. The direct Coombs test is used when immune-mediated hemolytic anemia is clinically suspected. Direct Coombs test detects antibodies or complement fractions on the red blood cell's surface. We can use it to screen many diseases like the hemolytic diseases of the newborn, warm antibody autoimmune hemolytic anemia, and systemic lupus erythematosus. In all these diseases, antibodies are responsible of destruction of cells by fixing specific membrane antigens. Human antibodies can fix specific antigens at the surface of different cells and then cause their destruction by complement, ADCC, opsonization, and other mechanisms. In autoimmune hemolytic anemia, antibodies fix antigens on red blood cells' surface, then cause their chronic destruction, which reduces their number and causes anemia. In fetal-maternal alloimmunization, which we will detail in other videos, mother's anti-D antibodies fix on fetal red blood cells surface on the antigen D and cause their lysis, which is responsible of jaundice and neonatal anemia. Coombs test is used in diagnosis of these antibodies-mediated hemolysis. The purpose of direct Coombs test is to prove the presence of antibodies on the surface of red blood cells. It's called direct because we directly detect antibodies which are already fixed on antigens by adding the reagent. No more steps are needed. So, for example, we are going to take fetal red blood cells from the umbilical cord and realize a Coombs test to prove a maternal-fetal immunization mediated by anti-D antibodies. We put fetal red blood cells in contact with Coombs reagent. The presence of anti-D at the surface of red blood cells will allow to antiglobulins to fix them and then to agglutinate red blood cells, which will create a small blood clot that is visible. We call this hemagglutination, and the direct Coombs test is then positive. The absence of anti-D, that means the absence of maternal-fetal immunization, won't allow the creation of the blood clot. The direct Coombs test is then negative. We can also use a Coombs reagent that reacts with fractions of complement. We can get it by introducing human complement proteins in animals, exactly as we did with human antibodies. Let's now talk about the indirect Coombs test. Indirect Coombs test proves the presence of serum antibodies that are able to react with known antigens. These antigens are fixed on cells like red blood cells. It's used to prevent alloimmunization in transfusion and in prenatal screening of maternal-fetal lysis immunization. It's called an indirect test because immunoglobulins we are looking for aren't linked to antigens. They are free in serum. So we have to add new red blood cells, let the antibodies bind to antigens, and then add the Coombs reagent. We take blood from a mother or from a patient who will receive transfusion of red blood cells. Then we separate the serum that contains proteins and antibodies. We put this serum in contact with the new red blood cells that are totally different and independent from giver and receiver, but that contain the antigen we are looking for. For example, if we want to prove a rhesus phytomaternal immunization, we will use red blood cells that are rhesus positive, which contain the antigen D in their membrane surface, but we take them from a third person that is different from giver and receiver. We don't take them from the mother and we don't take them from fetus. We complete the experience by adding the Coombs reagent to the mixture. If the serum contains anti-D antibodies, they will bind to the red blood cells and the reagent will fix them. So, we will have a visible hemagglutination. If not, there won't be hemagglutination. As a result, hemagglutination proves the presence of anti-D antibodies in the serum of the mother because the hemagglutination can't occur without them.